Ladies and gentlemen, it's absolutely wonderful to welcome you to the Royal Society of Medicine this afternoon. I am so thrilled that we've managed to reschedule this after many train strikes, tube strikes in the autumn, and that Roger Barker has most generously been able to join us from Cambridge. I have to say, Roger has been I think he was the first neurologist I met, which would have been back in 2005, when the charity was just very first coming into being. And Roger very generously has provided unending, unstinting support and advice to us in our fledgling years and as we've grown. Roger has just been absolutely a phenomenal source of advice. And I seem to remember was at our very first research meeting in the vestry of the church in St. Clement's, which is where we were based in our very early years. So Roger, I am absolutely delighted that you're here. For those of you who are unfamiliar with the RSM, there are two entrances to this. This is my EasyJet bit, by the way. And for those of you online, this really does have absolutely no relevance whatsoever. But for the doors are to manual, and you go out on either side, and that will take you back out into the lobby, and the loos are downstairs. Please do make yourselves at home in this building, because it's very important, particularly for people who feel they need to stretch their legs from time to time. So without further ado, I am absolutely delighted that Roger Barker is going to join us now and talk about cell replacement therapies for Parkinson's. There are 400 people online, so we will be needing to use microphones for any questions and any answers as they go along. Also, just to point out two of my very important colleagues here, we have Simon Stott, who is our Director of Research, and Richard Wise, who's our Director of Clinical Research, sitting up here, just so I'm not on my own. Roger, over to you.
Well, thank you very much. It's very nice and very kind of you to invite me. It's nice to see Richard in the front row. Last time I was here, he was on the stage doing his Desert Island Discs in celebration of his particular birthday and the amazing success of the rose trees in funding research over many years. So I'm not quite sure. I just pressed this button. Oh, that's right. Okay. There seems lots of things up on the screen here. So what I thought I would do today is I would take you through a bit of a history trip of cell-based therapies for Parkinson's disease, particularly around the idea of dopamine cell-based therapies. So not other types of cell-based therapies, which I will mention as I walk through this. And I thought the way to sort of start with this was to sort of explain the principles of what it is that we're trying to do and what we're not going to do. So I think this is an area where people slightly misunderstand the idea of what cell-based therapies for Parkinson's disease are really all about. So this is about repairing the brain. And so the first thing you have to ask yourself, well, what actually do I mean by repairing the brain? So of course, everything we do at the moment in terms of Parkinson's disease is not trying to repair anything. You're simply trying to replace things that are missing, either the missing chemicals in the form of drugs, or to deal with dysfunctional pathways by correcting them back to something that's normal or stop them being abnormal. So repair is you're actually trying to mend it. So we're actually going to try, and that's what I'm going to talk about this afternoon, we're going to try and repair a part of the brain in Parkinson's disease using cells. So then the first question is, well, we say that's what repair is, but what are we actually going to repair? So what part of the Parkinsonian brain are we going to repair? Now, as many of you will know, this condition is much more complicated than we originally thought. That the pathology of this condition is much more extensive than was originally thought to be the case. So this is a well-known slide of the so I'm sorry for people at home, because they're not going to be able to see my pointer, I don't think. But anyway, you here have the privilege of seeing the pointer on the screen. So this is the pathology which you see in Parkinson's disease as defined by a neuropathologist in Germany 20 years ago, Piper Braat, which is that the pathology starts in the lower parts of the brainstem and then it ascends up through the brain. And this bit here, which is at the top of the brainstem, which is called the midbrain and the substantia nigra, is where the dopamine cells are lost. But you can instantly see from that picture that there is extensive pathology all over the central nervous system. There's probably pathology outside of the central nervous system. So if I'm going to repair the brain or the body in pod disease, in essence, I would have to repair all of that if I'm going to repair the entire system. So I can't do that. So I'm only going to repair one bit of it. And the bit I'm going to repair is this bit called the nigrostriatal pathway, which most people are familiar with. So at the bottom top of your brain stem, which lies just at the top of your neck, at the top of that is the midbrain, and this is the midbrain cut across. I feel a bit, a lot of people here know a lot more about this than me, but this is the pigmented cells of the dopamine cells, which project up from this part of the brainstem up to a thing called the striatum. It's just important to remember the striatum, because that's where dopamine works, and that's where we've been trying to target our therapies. So in Parkinson's disease, you lose these dopaminergic nerve cells, as you are well aware. And in the normal, healthy human brain, we have 500,000 dopamine cells on either side of our brain. And when we lose half of those, so about 250,000, you present with the very first motor features of Parkinson's disease. So in essence, if I can put about 250,000 dopamine cells into that system, I should, in essence, be able to put it back to normal. Now, that might seem like quite a lot, 250,000, but compared to the 90 or 100 billion cells, or however many you have in your brain, that is a feasible number. So what we're trying to do is we're trying to repair one pathway. We're trying to repair and replace this lost dopaminergic projection. Now, why would that be a useful place to start? Do we have any evidence to suggest that targeting that pathway would be helpful? Well, I think we have a lot of evidence to suggest that going for this pathway with a cell-based reparative strategy would have major impacts. The first thing, and this is also important to understand the restrictions of what we can do, as well as some of the advantages of it, all we're ever going to be doing with this therapy is we're going to be treating those parts of Parkinson's which respond to dopamine, which is obviously the mainstay of the treatment at the moment. So as you know, the treatments for Parkinson's are essentially all around dopamine replacement. So you either stop the breakdown of dopamine with inhibitors, you replace dopamine with L-DOPA and Sinemet or Madapar, or you stimulate where dopamine works on the receptors with dopamine agonists. You can also use infusions of L-DOPA in the form of Duodopa, or infusions of apomorphine, which stimulate the dopamine receptor. And then amantadine and deep brain stimulation are used to treat the complications which you see from the chronic treatment with dopaminergic medication. Now, as most of you will know, when you treat people with Parkinson's in the early stages, the response is very dramatic. And when the right dose is achieved, a lot of people essentially look as though they've gone back to normal. That is the best we can achieve with a dopamine cell-based therapy. We will only be able to do as well as you do with dopamine drugs, because obviously what we're doing here is we're simply taking the oral drugs and converting them into a nerve cell to replace the dopamine in the brain. So it should, if it works, help with all of the dopa responsive aspects of the disease. In addition, if it worked, it would essentially mean that everything we do currently in Parkinson's would be redundant. That's not to say that there are not other treatments you need to give, but the whole strategy around using dopaminergic medications and then treatments to treat complications of that would become redundant, because we corrected that aspect of the disease. So it has major implications in terms of what we would do treatment. We understand what it would treat in terms of the aspects of the disease. And not only that, but it would normalize networks throughout the brain. So if you actually put dopamine back to where it's lost in Parkinson's with cells in a nerve form, it should restore networks back to normal, because studies have been done where if you take people with Parkinson's and look at the connectivity of their brain and activation of bits of the brain before and after they take their medication, the drug normalizes the networks. The other advantage of this approach is that it will avoid the off-target problems you see with medication. So what do I mean by that? Well, people who take dopamine agonists, so ropinirole, pramipexole, these type of drugs can end up developing behavioral problems, gambling and such like. Some of the drugs drive hallucinations and confusion. That's because the drugs stimulate dopamine systems which are less severely affected. By using a targeted approach, replacing dopamine where it's lost primarily with cells, you would avoid all those off-target effects. So you wouldn't get the side effects that you see with the medication, including some of the complications which happen, like dropping blood pressure and such like. So it would be a much cleaner way to treat it. And the other problem it would solve if it works would be that dopamine is being replaced in the form of nerve cells. They will release dopamine in a normal fashion. And that will avoid the long-term complications you see with the L-DOPA drugs, which is that over time, as you will become aware, the tablets don't work or they start to produce dyskinesias, these involuntary movements with chronic use. And that is thought to be related to the fact that when you take the tablets don't stimulate the system in the normal way. Here we're putting back normal nerve cells, so they'll stimulate the system in the normal way, so you won't develop the complications from the oral medication. So there are huge potential for this therapy to change the natural history of treated Parkinson's disease, but it's also important to realize what you won't be doing. So a lot of people go around saying that this, this is a cure. If you have this therapy, you'll be cured of it. Well, it obviously won't treat anything that lies outside of the pathway, because I'm only repairing one pathway. I think that pathway is critical, and I've got lots of evidence to say it is because all the drugs work so well. But it will not treat all the problems outside of that pathway, which cause all sorts of other issues with respect to Parkinson's disease. So that means we do need to find disease-modifying therapies, which of course can be used with these type of cell therapies. But we won't be treating any of those. We'll avoid the exacerbation of those with the drugs if it works through the cell therapy, but we won't be treating that. We will not be curing Parkinson's disease. So I know it's cure Parkinson's, but I won't be curing it with this. And the reason being is that the disease process will carry on. So the accumulation of the protein, the alpha-synuclein, which drives the disease, will carry on because I'm not targeting that. I'm not dealing with that. What I'm doing is saying it's caused those cells to die. I'm just going to replace them. But as I've said, it could be used with other disease-modifying therapies. So people are aware of things like exenatide, we're going to hear about, and Broxol later on. Now, if those drugs work to slow down the disease, there's no reason you couldn't put cells into somebody's brain to replace the dopamine system when they first present, put them onto disease-modifying therapy, and use a combination of the two. And finally, this is not going to be suitable for everybody. So the people who this is going to be most suitable for are people who tend to be a bit younger, who have major problems around the dopamine system, who've had a good response to dopamine therapies. So for the moment, it's probably going to be people who are currently being considered for deep brain stimulation and such like. Obviously, as we move forward, if it really worked, then it could become first-line treatment for a lot more people, but we've got quite a way to go before we can say that. So I've tried to explain what we're trying to do. We're trying to repair a specific part of the brain, the loss of this nigrostriatal dopaminergic system. We're trying to put back dopamine cells to repair that bit of the network. We know what it will do. We have an ambition on what it will achieve, and we know what it won't do. So the next question is, well, if I want to repair that pathway, how am I going to do it? And I've already talked about the main way I'm going to talk about that, which is replacing cells, but you can think of it in terms of other ways. So you could talk about trying to repair from within. So could I get the remaining dopaminergic cells and fibers in the nigrostriatal system in the brain of people with Parkinson's to regenerate? So could I make, and I'm not going to talk about this, but I'm happy to take questions on it. Could I get more dopamine cells made in the brain to replace those that are lost as the disease process? Now, obviously, that process, if it did exist, isn't very efficient because you wouldn't have Parkinson's in the first place because they would keep being replaced. So enhanced neurogenesis, could I make more dopamine cells inside the brain? I don't think there's much evidence for that, but I'll come back to briefly talk about that. Could I make dopamine producing nerve cells directly in the brain? And I'll come back to talk about that at the end. Or could I regrow the system? Could I put some fertilizer into the brain to encourage those dopamine cells and fibers that are there to grow more efficiently? So all of those are designed to repair that dopaminergic system, but you're using what's there already to repair it. So that's what I mean by repair from within, whereas repair from without is a much simpler strategy where the idea is simply you want to replace the lost dopamine cells by putting in a new supply of those dopamine cells. And on that point, it's very important to realize that you can't simply replace the dopamine cells in the brain with any dopamine cell. If they're really going to work, you have to replace them with the dopamine cells that are lost in Parkinson's disease, which are the dopamine cells, which is in this structure called the substantia nigra, as I've already mentioned, which are called the A9 dopamine cells. If you don't use A9 dopamine cells, you ain't going to get such a big result. But And that is what I'm going to talk about now. But obviously, over the years, people have considered this in slightly different ways. And that's where I'm going to walk us through now. So when you're considering what dopamine cells to use, I've already said that the ideal ones are the A9, the nigral dopamine cells, which are lost in Parkinson's, but that raises quite a major question of where you're going to get those from. And this field began back in the 1970s and 1980s, when the access to human tissue, the ability to make stem cells, the ability to make dopamine cells was in a very primitive state. So what people did was they sort of said, well, where can we start looking for dopamine cells to use in the brain to replace those that are lost in Parkinson's? So the first place where people started looking was to say, well, where else do you make dopamine in your body? Can't we just whip it out from wherever else you make it and shove it in your head? And so pretty straightforward approach. So you go looking around and people looked everywhere. So the adrenal medulla, the little adrenal gland that sits next to your kidney, in the middle of that, the medulla, which produces your adrenaline, noradrenaline, produces rather tiny amounts of dopamine. That was thought to be a possibility. And based on almost relatively little preclinical data, that went to a clinical trial, and hundreds of patients had those transplants, a lot in the States, but some in the UK during the late 1980s and 1990s. The carotid body, which is dopamine cells that sit near your carotid artery in your neck, which sends oxygen, have also been used as a source of dopamine. But none of those actually make the A9 dopamine cells. An alternative source, and I'll come back to where we got to with each of these in a minute, is to say, well, actually, rather than look for a dopamine cell in your body and move it from one site into your brain, can't we collect the dopamine cells from aborted fetuses, the developing dopamine cells? So in termination of pregnancies, the tissue is collected, the dopamine cells are developing in the midbrain of that fetus. You take out those dopamine cells and you transplant those into the brain, and they will then grow and mature into the normal dopamine cells, which we have in our brains. So would that be the way forward? So you repair the brain using human fetal material. And obviously, there are many ethical issues associated with that. A third option, which only became really possible, I would say, in sort of 1998, but really only possible in 2011 and 12, was the idea of making dopamine cells from a stem cell source. So a stem cell is a cell that divides, so it produces its own self, so they can carry on dividing forever, and then they differentiate. So every single person in this room at one point was an embryonic stem cell. So they give rise to every cell in your body. And in 1998, Jamie Thompson made the first human embryonic stem cell lines. They come from spare embryos from IVF programs. And the idea there is you get make a cell line from an embryo, and then you have endless supply of cells, and you turn those into the dopamine cells you need for Parkinson's disease. And then the final source is the idea that you actually take an adult cell, so whether it be a skin cell, a fibroblast, or a blood cell, and then you reprogram that adult cell back to a stem cell state. So these are called induced pluripotent stem cells. Shinya Yamanaka was the person who led the work on this in mice in 2006 and humans in 2007. So I think it's quite important to remember iPS cells. If I was giving this talk in 2006, I would not be talking about iPS cells, and no one would have a clue what we were talking about. So they are still relatively new, but the idea of this is you can take adult cells, reprogram them back to stem cells, and then turn them into dopamine cells. And obviously that raises the possibility of using your own cells to repair your own body. So people are quite excited by that, but there are some fairly serious issues around that. So where have we got to with all of this? So the first thing I will say is that whilst the idea of using adult tissue was intuitively quite attractive, it proved to actually be fairly useless. And the reason was that these cells produce very little dopamine. They are adult cells who do not like being moved around. Like a lot of people, as you get a bit older, you don't like being moved around. And they did not like being moved around. So they were transplanted into people's brains. I said hundreds of people had this. And essentially what it showed was it didn't work. The cells did not survive. And there was no real benefit from it. So by the mid-1990s, this whole field using adult cells had essentially disappeared. And part of that was the failure of the clinical trials to produce any benefit in the face of positive data that was starting to emerge with the fetal dopamine cell work. So this work has its origins back in Sweden. about whatever it is now, about 40 odd years ago. And essentially, this study, which began all of this work, was done not to repair the brain. So the original work was designed to try and understand how cells normally develop. So the idea of the work was you took the fetal cells, you put them into an adult brain, and you just wanted to see how they normally grow. Actually, they then realized, because the adrenal tissue was being used to try and repair the brain pulse disease, wouldn't this be a smart way of trying to repair the brain in pulse disease, to use these fetal tissue? So this is how it works in the lab. And the reason I'm going to emphasize this is we have enormous problems in trying to translate from the lab to the clinic, because the only animal on the planet that gets Parkinson's is us. As a consequence of which, we don't know quite why humans uniquely get it. It doesn't seem that non-human primates get it. As far as we know, tortoises don't get it, but because they live very long. But I'm not quite sure what a Parkinsonian tortoise would look like, to be honest. But it's a problem. So everything we do in the lab is an imitation of what we see in the clinic. And therefore, if it works in the lab, it doesn't necessarily mean it will work in the clinic. And conversely, if it doesn't work in the lab, doesn't mean it might not work in the clinic. So it does make a problem with the preclinical work. But essentially, the way that people have tried to study this in cell replacement around dopamine is actually to do a very crude model of Parkinson's. So essentially what you do is you knock out the dopamine pathway. So with toxins or whatever, you kill off the dopamine pathway in the rat brain. So this is a rat brain. This is the substantia nigra, projects up to this thing here, which is the striatum. So you kill those fibers by toxins, or you can make models of them. You then monitor some deficit, which can be either motor or thinking. You then put your cells into where the dopamine normally works. If you put them in the nigra, they live there quite happily, but they don't project up. And this is where it has to work. So dopamine has to work. The cells have to be placed where dopamine normally works in the striatum. So all of the work I'm going to talk about is putting cells not where they're lost, but where they project to and where they work. So you then transplant the animals with these cells, you monitor the deficits, and then you sacrifice and you look at the brain. So all of this work was done over an 8-year period. And importantly, it was done in many different labs by many different groups who all found the same thing, which means it's likely to be correct. So if you read something which is all the preserve of one lab and only one lab can get it to work, a question mark just exists as to exactly how reproducible and robust that is to be taken out into other places, namely the clinic. So what we found by doing all of these experiments in rats and mice, and this is a transplant I did in 1993, when I first started doing research into this. So this is a rat that's had its dopamine fibers killed. All of what you see on here, all of this brown are dopamine nerve cells on here, and these are the fibers that grow out to repair the brain. And what we found through all of these experiments during the 1980s was that allografts, which means tissue from the same species, so it would, in our case, that's human to human. Autologous is from you to you. Xeno is from one animal to another. So if I was to do pig into us, that's xeno. If you have a kidney transplant, that's an allo. So the allografts survive long term, which in a rat or a mouse is a year. Ain't very long in a person's life, but that's the best we could do. So we could show that the cells survived, that they turned into the dopamine cells, as I show you here, that they sent out connections. So they made connections to and from the brain. They released dopamine. They made the animals better, and it could be reproducibly done in many labs. So you sort of think, okay, that's great. I mean, is it absolutely extraordinary that it worked? Because essentially what you're doing here is you're collecting dead fetal material, you're shoving it into an adult brain, and it not only survives, but it turns into what it was destined to turn into. It makes connections, receives connections, and makes things better. And if someone came to you that as a sort of research idea, you'd say, I think that is pretty crazy. But it worked. And it was on the back of that clinical trials then started in Lund in Sweden in the late 1980s, 87, and in other places around the world. And I'm not going to go through all of those trials, but I will show you what benefits you can see when you do this. So over the course of the next sort of 15, 16 years, the best benefits you could see with this. So this was now translated into the human situation. So the idea here is you take someone now, a person with Parkinson's, typically had the disease 10 or so years. You collect tissue from elective termination of pregnancies. So when we decided to terminate pregnancy, you collect the fetal tissue. You need to collect quite a lot of fetal tissue in order to transplant one side of the brain. You collect that tissue, you turn it into a sort of cell suspension, you inject it into the brain of the person with Parkinson's disease, and you immunosuppress them for a period of time, and then you wait and see what happens. And in the best case scenario, so these were two patients from London who were transplanted in Sweden. What you can see here is these patients had Parkinson's disease for 10 years by the time they presented. These are their scores. The higher you are, the worse you are. So this patient here, pretty bad Parkinson's, this person here, not too bad. They then had one transplant, one side of their brain grafted, and a few years later, they had this other side. And on this side, they had the same. And what you can see is that over a 5-year period, their Parkinson's improved. So by here, they were now at a level which was probably the score they had when they first presented to their doctor, which would now be 15 years previously. And that was then maintained for the next 15 years. So here we are now 30 years into Parkinson's. And at this stage, they are on no medication, both of these patients, and they have a motor score that is less than when they first presented. So 3 decades into the condition, in these two patients, you had essentially restored many aspects of their disease back to normal. And that correlated with a restoration of dopamine in the brain. So this is a scan where you look at dopamine in the brain. This is a thing called the caudate, and this is the putamen. This makes the striatum. This is the area that gets the biggest hit in Parkinson's disease. So if you ever have DAT scans, you lose this comma. And what you can see here is that there's a restoration of dopamine pretty much back to normal. So what these early studies showed was that in certain patients, it was transformative in terms of what happened. You put them back to normal, more or less. They stopped their medication, and that was maintained over decades. One of the patients who had a transplant on one side of their brain in 1989, who then died 24 years later in his mid-80s, his brain was then collected and was looked at pathologically. This side had no transplant, and there is nothing to be seen there. This brown smudge, which you see throughout, are all the surviving cells that were put in a quarter of a century earlier. So these dopamine cells have survived. So one of the questions when people began was, we don't know what causes Parkinson's disease. Perhaps it's the brain that's bad for cells and it kills them off. So would it kill off the cells? There's some evidence that it may do, but at a very low rate. But what you could see here was that this patient, who was also as a Swedish patient, who a number of years after his transplant came off all his medication, actually had a quarter of a century later surviving dopamine cells in the brain with innervation, which looked normal. So you could actually repair the brain with clinical improvement and restoration of dopamine levels to normal. And not only did it just improve things which we measure, because in those days, the idea that the patients could contribute anything in terms of what would be a useful measure was not something that people were particularly attracted towards. We had our measures. And of course, one of the things that people would say is, well, that's terrific. Your UPDRS gets better. I feel like rubbish. I mean, I don't care. My measures are better. So this is quality of life. And it shows that patients' quality of life improved with it. So it didn't just improve scans. It didn't just improve these arbitrary measures, but it actually made people feel better. And more than that, so this patient whose brain you're looking at here, 15 years earlier was the subject of a scanning study done in London at the Hammersmith Hospital. His transplant was shown to release dopamine in a normal fashion. So and the idea was they stimulated the transplant with amphetamine. It released dopamine just like the normal brain releases dopamine. So what all of this shows is that you could, in certain patients, get long-term clinical benefits with restoration of dopamine levels in the brain to normal, with evidence that it survives long-term at post-mortem, improvements in quality of life and non-motor measures, and that the transplant repaired that brain and released dopamine in a normal fashion. So if that's also good, I'm now 20 years further on, and this doesn't seem to have got anywhere. So why is this? Well, the problem came that in 1992, if you see it as a problem, there was a change in the White House in America. So Bill Clinton came into the White House, and he allowed federal funding for work with human fetal tissue. And through the National Institute of Health in America, they funded 2 double-blind placebo-controlled trials, which were then published in 2001 and 2003, and pretty much killed the field for many years. And why was that? So these are the 2 trials that were published. So in the first trial, which involved 40 patients, the idea here was that 40 people were recruited to the trial. All 40 people went to theatre. All 40 people had someone play in their head. In 20 of them, they drilled through the skull and they put cells in the brain. In the other 20, they just pretended to do that and made a dent in your skull. So when you're the patient saying, did I have the operation? Oh yeah, there's a hole in my skull. So someone's obviously been there. And then at the end of a year, they were asked if they felt any better. That was the trial. And the result was they weren't any better. People didn't feel any better. And the slightly worrying thing was that some of the patients had now started to develop the dyskinesias, which you see in people who take chronic L-Dopa treatment, but now in the presence of a transplant and the absence of medication, which is a bit of a problem. Because I obviously can get rid of anyone's L-Dopa-induced dyskinesias, because I can just stop them taking L-Dopa. They might not want me to, but I can get rid of them. If you've got a transplant in your head and you've got involuntary movements, I can't get the transplant back. So now we have a bit of a problem. They weren't bad in the majority of cases, but nevertheless, there was no benefit and there were side effects. I mean, interestingly, when they showed the patients the video of what they looked like before they had the operation and asked them a year afterwards of what they felt, they actually thought they were better having had seen what they were like before, which I think is typical for all of us, really. We can never quite remember what we're like last year. And sometimes when you're reminded, you think things are slightly different. But essentially, this trial was negative. Didn't work. Didn't work. And there were side effects. You think, okay, well, there were funny things they did in that study. And then the second study, which was published in 2003, was a slightly different study. So here there were 34 patients. A third had a small transplant, so they had a little bit of tissue put in their brain. A third had a loss of tissue put in their brain, which is what the 4 donors refers to. And 4, they just had pretend transplants. So it's very controversial, this, because this is imitation surgery. I mean, people go to theatre, they have general anaesthetics, and in this case, everybody gets 6 months of cyclosporin, so immunotherapy. And the upshot of this study was it didn't work. So they waited 2 years here. They used this thing called the UPDRS, and what you can see is all the groups here ended up exactly the same. And now half the patients had developed these involuntary movements. So essentially, this looked as though it was a waste of time. It didn't work if you did the studies properly. Now, of course, in the background of these patients in these open label studies where they know what they've had, have clearly done very well. So on one hand, you say this is the right answer, and all the other stuff you've shown me is just placebo, investigator bias. But it's very hard to get away from the post-mortem and the imaging. You can't really put that on. Or there's something wrong with these trials, or things aren't quite as they seem. One thing I will just point out here is that the patients who had the transplants at 6 months had all improved. At 6 months, their immunotherapy to stop the transplant being rejected was discontinued. And of course, maybe it wasn't going to work anyway, but it is curious that the patients at 6 months then slowly got worse. These patients, in contrast, had no immune suppression at all. They were thought that they didn't need it, so they were given no immunosuppressive drugs at all. So here we are in 2003, and we're sort of at a bit of a sort of difficult situation, because essentially this finished the field. So in 2000, if I was giving this talk in 2003, I would say, well, there we go. That was a nice little adventure in Parkinson's disease. We're going to have to go away and think of something else. Now, I decided that actually I didn't think we would do that. And my very good friend, Anders Björklund in Sweden, who invented this whole field, we thought we ought to just revisit it before we abandoned it. So what we decided to do was to try and work out whether there was any merit in this approach which we could take forward in a new trial. So we did this sort of rather limited meta-analysis where we tried to collect all of the data we could on all of the trials that had taken place in those 2 studies I've shown you, the studies which have been taking place in Sweden, as well as other studies around Europe and in Canada. And what we did was we plotted all of the patients in these trials. So this is the change, percentage change in your score from baseline 2 years after you had an implant of one of these cells. And essentially what you could see was that about a third of the patients 2 years post-transplantation had a significant improvement in their Parkinsonian state. Obviously, 2/3 did not, and some patients did particularly badly. So then we tried to analyze the data and say, well, what is it about the patients who've done well? Is there anything we can learn about the patients who seem to be optimally responsive to this? And what we came up with was that patients who were younger and less advanced seemed to be the people who did best, especially people who didn't have a lot of L-DOPA-induced dyskinesias. They seemed to do much better. There was clearly about the amount of tissue you used and how you put it in that was critical. So different studies had used different approaches. Immunosuppression, which I've already mentioned, seemed to be important. The more aggressive the immunotherapy, the better the outcome. And the longer the follow-up was also important, because as I showed you with that slide with the patients who improved over many years, it took several years for this to improve. And if you had gone at 6 months or 12 months, you might well have missed the advantage of what the transplant was going to do if you'd waited longer. So it was on the back of this that we went to the European Union in the days when you could go to the European Union and ask for funding, which as of Tuesday of this week, I don't think we're allowed to do. And they bravely funded a new trial, because the data when this trial was approved in 2010 was very much against this. So this was the TransEuro study. And essentially the idea here was to optimize the type of patients based on that meta-analysis and other work we'd done, standardize how we prepare and deliver the tissue. We would do this in 150 patients. The idea was to randomly select 20 patients, do an open label study, i.e. everybody knows what's happening, look at the data a couple of years later, and then do this double-blind placebo-controlled trial with sham surgery and everything. That, I mean, massively ambitious and almost impossible to deliver on, I would say. And that is exactly what we then demonstrated, that it was impossible to deliver on. And what we managed to do was one incomplete transplant trial, not over 5 years, but over 11 years. So we ended up grafting 11 or 10.5 patients, and we waited 3 years. So we finished just the year before last with the final follow-up on our final patient. Now, we were very fortunate enough to get funding to continue with this, and I would like to thank Parkinson's who helped us with that bridging funding. And there were many reasons for why this was so delayed, around devices and changes in legislation and changes in regulation, all of which meant we had to keep changing everything we did. But the fundamental problem for us was that for these 11 patients we grafted, 10.5 patients, that's 21 transplants we did, there were another 87 surgical slots that we had to give up because we could not get enough fetal tissue to graft the patient. So in order to transplant one side of the brain, you need at least 3 to 4 fetuses collect over 3 to 4 days. So if I'd had a stem cell source of dopamine cells when I began this trial, which did not exist in 2010, in the time it took me to graft 10.5, I could have grafted, in essence, 21 plus 87, 108 patients. So what this meant was that this whole approach is clearly not viable. So we've got to go and find some stem cells if we're going to make this a viable treatment for other people. So that's where we started in 2011 to take this forward into the new generation where we are now. So in 2011 and 12, there were 2 very important papers which came about. One from a team in New York, Lorenz Studer and Vivian Tabar, who managed to demonstrate that you could take human embryonic stem cells and turn them into A9 dopamine cells of the type lost in Parkinson's disease. You could transplant them and they worked. And my very good colleague and friend, Marlin Palmer and Agneta Kirkeby in Lund, who managed to do exactly the same thing using a slightly different protocol. So now, 10 years ago, we suddenly had the potential to take embryonic stem cells and turn them into the dopamine cells that we wanted for use in Parkinson's disease. And in Japan, where they had discovered or made, or whatever you want to call it, the first human iPS cells, Jun Takahashi managed to show that he could take human iPS cells. So he could take skin cells from either people with Parkinson's or from people without Parkinson's, turn them into iPS cells, turn them into dopamine cells, transplant them into monkeys, and then demonstrate that they survive long-term with functional benefits. And so when that paper was published in 2017, he was then able, the following year, to announce that he could now go to a clinical trial where he could transplant patients with his iPS-derived dopamine cells. And so that trial began in 2018, and as far as I know, has now completed grafting 7 patients, and the results are keenly awaited. It's obviously taken longer than we thought because COVID came along and put a massive hole in this program for 2 years. During this period of time, another group in America have actually published a single case. And I think this is a bit of a sort of warning about where you have to be a bit careful with by looking at the data. So this was published in the top medical journal on the planet, the New England Journal of Medicine. It was a single patient, and what this patient did was they donated their skin to be turned into iPS cells, which was turned into a dopamine cell, which was then put into their brain. So this was autologous. The patient provided their own cells for grafting into their own brain. And then the preclinical data showed that they could do this, but the preclinical data was not overwhelming in terms of what you saw. So the cells didn't look fantastically healthy, but nevertheless, it seemed to do what they wanted it to do. So they went on and grafted this patient. They made iPS cells, turned them into the A9 dopamine cells, transplanted one side of the brain, and then transplanted the other side of the brain. And what they found was that it made absolutely no difference in terms of the imaging. So there was no evidence from this that those cells had survived. There was no evidence on an objective measure that the patient is actually better, but the patient felt fantastic. So this quality of life had gone from really bad to essentially back to normal. Why that is, one could leave for speculation, but this would demonstrate that whilst it was safe, it didn't really show that they had worked. So I think whilst people have cited this as a very useful paper in terms of safety and efficacy, I would say it shows that it's safe, but efficacy we've yet to see. So where we've got to with this work is that over the last 11 years, we have tried to take what was a laboratory-based approach for making dopamine cells from ES cells to the level that's needed for a clinical trial. And what has been demonstrated both by the group of Lorenz Studer and the group of Marlin Palmer and other groups is that this is highly reproducible. So many labs can do this. We do it all the time in Cambridge with the protocol that they developed in Lund. So we make dopamine cells all the time from embryonic stem cells. So it's very reproducible. It's incredibly efficient. So if I start with a 6-well plate of ES cells, so if I start with that, then by the middle of March, I would have made enough dopamine precursor cells to treat 300 people. So so if I had 2 plates, I could treat everyone who is actually watching on this and here with the dopamine cells that I could make. So that's great because it's highly efficient. That has 2 advantages. If it's highly efficient, it means that the chances of it going wrong are small. Secondly, it's very cheap because you have to do all this in special clinical facilities. So that costs a lot of money. So suddenly in 16 days, I can make enough dopamine cells for 300 people with 1 plate. If I have a robot and 1,000 plates, I can make enough dopamine cells to treat the whole of Europe. That has financial implications, which people have suddenly woken up to. So it's highly efficient, highly reproducible. And what we've managed to work out, we in the Royal Sense, in that other people have done this, is we've got markers which are predictive when we take the cells out of the freezer before we put them into people's heads, that they are going to turn into what we think they're going to turn into. Because obviously what we're not putting in people's brains are mature dopamine cells. We're putting in cells that we want to become dopamine cells, so they're precursors. So it's like a prodrug. When you give someone a drug that gets converted into the active drug, this is what we're doing here. But we have to know that when we take the vial out of the freezer, it will do what we think it is. So we've had predictive markers. And as I say, we have used, so this is one ES cell line, H9, which is actually the one that's been used in the American trial. This is the one we're using in our trial from Roslin cells in Edinburgh. And this is a new line which was made in the Karolinska. All of them with this protocol can be turned into effective dopamine cells, which reproducibly treat animals, which I talked about earlier, back to normal. And it's highly, highly efficient. And the final thing is that what's been demonstrated in the group in Lund is that when you use these cells and compare them to human fetal transplants, which have this long history in patients, they look very similar. They behave in the same way. So if I put 300 of these ES-derived dopamine cells into a brain and compare it to 300 human fetal dopamine cells from an aborted fetus, they look and behave the same. So we've probably produced something that pretty much looks like the thing that we need. So this has now led to trials. Not only has it led to trials, it has led to massive investment. So when we began Transgero, I could be pretty confident that the world investment in cell-based therapies for Parkinson's was approximately $0. And now it's billions. And the reason is that people have, oh gosh, this has changed color in transferring. The reason is this highly efficient process means that you could charge quite a lot for this, but it doesn't cost much to manufacture. And coming back to where I began, what you could actually do with these cells could transform how we treat Parkinson's disease. So just to run you through all the groups in the world that are doing this, this is the group that I was talking about in New York, the Vivian Tabar and Loren Studer. They set up a company called Blue Rock. They transplanted their first patient in May of 2021. They transplanted their last patient in their phase 1 study, phase 2A study last May. Their results will be out in the latter half, final quarter of this year. Blue Rock are now owned by Bayer, who are obviously a big company. We have set up our own trial, STEM-PD. First patient will be transplanted this month. And Novo Nordisk, fourth biggest pharma on the planet, are working with us to take that forward in their own study. That's the single case I talked about. Fujifilm, who've now set up their own company to do it in America. Aspen Neuroscience in California are doing it with patient-owned cells. And there are other trials. Sara is the group in Japan that I talked about. All of these are attached. That's attached to a company. That is a company. That's Fujifilm Cellular Dynamics. That's a company by Novo Nordisk and the International Stem Cell Corporation. So this has now attracted a lot of investment. And over the next few years, we're going to work out whether any of this will actually deliver in the way that I said. So in the last few minutes, I just wanted to say about the future. Where do I see this going? Because you could say, well, that's it. I mean, and that might be the case. If this works, why bother with anything else? But there are a few things that people are thinking about. I just thought I would introduce the ideas. So one, which I haven't really talked about, is that the pathology of Parkinson's is this aggregation of alpha-synuclein. And when you look at people who've had fetal dopamine transplants 10, 15, 20, 25 years after they've been put in, the transplanted cells have developed some of the pathology of Parkinson's disease. Which is very interesting because those dopamine cells are only very young, yet they've got the pathology of an age-related disease. So how is that? And that's raised this whole idea that this disease can spread from one cell to another and seed pathology. So this whole idea of alpha-synuclein spreading and causing disease began as a byproduct, if you like, of the fetal transplant work. Obviously, one way to stop that ever happening is to make a stem cell that has no alpha-synuclein. So if the stem cell has no alpha-synuclein, it cannot seed pathology in that cell because anything that gets into that cell from the patient says, I want to bind with your alpha-synuclein and turn it into a pathology, it won't find it. So people are now working on making alpha-synuclein knockout cells so that you could never get the disease in those cells. Another way of gene editing it, which is something that we're doing as well as other groups, is to say, I've sort of talked the fact you have to use immunosuppression, or we think you need to use immunosuppression because the body will recognize these cells as foreign. So could we actually make the cells less immunogenic? So you knock out things in the cell which are recognized by your immune system as foreign and kill it off. Can we make them less immunogenic? So people are after this thing called a universal cell where you knock everything out of the cell so the immune system cannot recognize it. So that is terrifically exciting. There is a problem with that, is if that cell goes rogue, you've got absolutely no way of dealing with it because your immune system will not see it at all. So these universal cells are very exciting, but they can create some safety issues. And they're probably more relevant for things outside the brain, which has some immune privilege, such as the heart, where people are trying to use stem cell cardiomyocytes or beta cells to make insulin and such like. But anyway, we're working on and have made some of these knockout cell lines with the hope that if we can make better cells which are less immunogenic, we could reduce the amount of immune therapy that people would need to take post-transplantation. The other area, which of course comes back to something I said right at the beginning, is rather than transplant in cells, given you could argue you've got all this clever technology, all these clever scientists worked out how I can take a stem cell and turn it into a dopamine cell. Can't I just take a cell in the brain, which you've got anyway? I mean, you've got lots of spare cells in your brain. Can't we just persuade them to turn into a dopamine cell? So people have been very excited with this. So the idea would be where the dopamine normally works in the striatum, you have your nerve cells, and then you have the supporting cells called the glial cells. And you say, why don't we just hijack 250,000 of those glial cells? You don't really need them all, and persuade them to become dopamine cells. Then I don't have to worry about rejection. There's no ethical problems. I've basically just converted one population of cells into the cell that I want. And so this has been tried now for 5 or 6 years. And so there's a group in the Karolinska that did this. And then there was this paper that appeared in Nature 3 years ago saying it looks pretty easy. We can do this pretty well in our animal models. So we basically put in a single factor. We converted astrocytes in the striatum into dopamine cells. Animals got better. Thank you very much. So that looked as though that was going to be a real runner. And then to their credit, Cell, which is probably one of the most prestigious scientific journals in the world, published a paper a year later saying that's all well and good, but we did exactly the same experiments as this paper in Nature and couldn't get it to work. And the reason why we think they found what they found was it's actually nothing to do with converting astrocytes to nerve cells. The dye and everything you were using to show that you'd converted it actually leaked out into the host nerve cells. So that's why they looked so much like the host nerve cells, was because they were actually the host nerve cells. And so this idea of in situ reprogramming is very attractive. People are still working on it, but currently we haven't got there. And then the final idea, which I just wanted to float before I conclude, was that could you improve your transplants in some ways? So could we actually get them to survive better and make better connections? And this comes back to another idea, which is to try and regrow your dopamine system with growth factors, GDNF. Many of you will be very familiar with that. But a group in Melbourne, Lachlan Thompson and Claire Parrish, have taken this on, where they've tried to combine therapies by putting in transplants and putting in growth factors to try and enhance the survival and the growth out of that transplant into the host brain. And this is looking very encouraging as a way that maybe you could take forward to improve what we're doing already. So to conclude, using cell therapies, dopamine cell therapies around the brain, what can we say? Well, I think there's a rationale for what we're doing. It's clear what we're trying to do and what we can achieve and what we can't achieve. And I think this is doable. 250,000 dopamine cells, that's what we've got to do to repair this part of the brain. And I think what you can say from the human fetal midbrain transplants is that when it works very well. It just doesn't work in everybody. And if I cannot get a consistent good response, then I ain't got a therapy. So if only 1 in 10 people show a fantastic response, it's a great starting point, but that's not a competitive therapy. And I think some of the reasons for this relate around patient selection and the fact that using human fetal tissue, you can never standardize it. It has lots of cells in there that you don't want. So most of what you transplant are not dopamine cells. It's what normally develops in that part of the brain. And that we now, I think, can move that on by using cells where we have a much more prescribed product at the end of it made from stem cells. But nevertheless, the human fetal dopamine transplant trials show that cells survive and can have dramatic effects. So this whole principle should, in theory, work if we can find an acceptable, reproducible, affordable cell-based therapy from stem cells. And that, I think, is, where we are now. These are now entering into trials. The great thing is that pharma's investing in this. The problem with this is pharma's investing in it. So there are 2 issues with this. One is the cost of this is huge. So to do one, so people always say, why don't you do autologous iPS cells? So if I wanted to treat these 3 beautiful people up here, and they all had an iPS-derived dopamine cell, the cost of manufacturing and testing that cell would be about 4 or 5 million per person. So you cannot do that with the current regulation, the current way it's doing. So in order to take this forward, so in order to take that 2012 paper from Marlin to a clinical trial now, has probably cost us, or is required somewhere in the region of 10 to 15 million pounds, I would think, in order to get to that point. Now, trying to do that piecemeal with grants is difficult. We're very grateful to people who've supported our work, because obviously we couldn't get there without it. But ultimately, to get beyond that, I can never go beyond my trial. The trial we're doing at the moment is costing 5 million. The next trial would cost 15, 20, 25 million. I need Big Pharma to support it. So without it, you're not going to get anywhere. The problem with Big Pharma is they want quick results, and they can drop programs just because they're not happy with things or there are problems elsewhere. And the big problem with this is the benefit from these therapies take years to become evident. They will not wait years. So they always say to me, when should we wait? When should we do a dose escalation change? And I'll say, well, let's wait 3 years. And they'll say, I'll give you 6 months. And so you have this tension between pharma wanting things to move quickly, but the necessity for them to help us. And whilst all this is going on, and then this ship sails with all these trials, which is where we are now, I think there are new things coming along. And the idea of direct reprogramming the brain, I think, is very exciting. And I suspect that that will be the next breakthrough, along with trying to actually have cells which are much more regulatable. So that's the area we're trying to think about now, is how we can make cells where we can control them in a much more sensitive way. So with that, I will finish. I mean, most of this has not been my work. This is our research group, which met last year on our retreat, the first time in 3 years. This was when the team in Lund, about the same time when we were setting up our STEM PD trial, all the people who funded us over the years and our various collaborators. But particularly what I would like to say is there are 2 groups of people who've made this possible. One is the team in Lund have been fantastic. And secondly, it's been the global community. So all of the people I've mentioned who've done all these studies, we meet every year to work out how we can work together to deliver this to the clinic. And that has made a huge difference. So I will finish there, and thank you for your attention.
Roger, thank you very, very much indeed. I am sure that there are questions both online but also in the room. Rosie, are you able to dive in with a microphone? You need to just bear with us so we can get microphones so that everybody online can also hear. I think that's working. Richard.
Hear me? Can you hear me? Yes.
Yes.
Roger, that was brilliant. I can say that because I'm an accountant and I think I understood everything you said. So we've been supporting research for over 30 years, and I suddenly remembered 30 years ago we were invited to support this project in Sweden with the stem cells, and it sounded like a good idea. Well, that was 30 years ago, and we still haven't got there yet. Can't hear me? Can you hear me?
Yes, that's better. Yeah. Thank you.
We supported research in stem cells in Sweden 30 years ago, or we invited to, and we haven't got there yet. So do we need another 30 years? Well, what do you see in your crystal ball for the future?
Well, you always say 5 years. That's the answer you always say.
I've learned that.
And then 5 years later, people said, you said 5 years ago, 5 years. I think the problem. So I mean, 30 years ago, we weren't working with stem cells as such. We were using fetal tissue. The big breakthrough came 11 years ago, 12 years ago, when we could make stem cells into dopamine cells. That was a big breakthrough. What's taken the time since then is there's a huge difference between making something in a lab that works and making something that's of a clinical grade. And that is an enormous amount of work and an enormous amount of money. I think we're on the cusp of it now. So the next, I mean, I'm going to say 5 years, but I mean, there are currently our trial, there'll be a Novo trial, there's an Aspen trial planned, Bayer again with their trials, Novo Nordisk again with their trials. In the next few years, we will know whether this therapy is going to work or not. But it does take a lot longer. So there's a great talk by my colleague, Alistair Coles, on Campath, which is a monoclonal antibody used for MS. And they got a license for that in 2012, having done the first trial in 1990. So 20 years is normally not unrealistic from an idea and getting a sort of lab and starting in the clinic to getting a license for it. So I'm sorry. Yeah.
Roger, I think we've got a question online. Suzanne, are you okay to?
Yeah. We have a number of questions actually directed here, and that is, what should individuals look out for when considering using private stem cell clinics? One person has actually asked about Swiss Medica, but they say there are a lot opening up now across Europe and Ireland, and would like your opinion on that, please.
Yeah, don't do it. I mean, there is a whole industry of stem cell tourism. So if you wanted to have a stem cell transplant for Parkinson's disease tomorrow, there are probably 300 or 400 clinics around the world that would give it to you. And my advice on stem cell therapies is there is no evidence that these work. That's why we're doing the trials and we're doing the work that we're doing. The rationale for what we're doing is we're replacing dopamine cells. So I've tried to explain that in the talk. A lot of these other stem cell therapies which are being advertised, they're not clear how they work. They're not necessarily put in your brain. So you will have sort of fat stem cells put under your skin, and that somehow will make you better. So, it's not to say that they couldn't work, but it's very hard to see how they could work. Anything that's not proven, you shouldn't be paying for. So if you want to go and have a private hip replacement, that's fine, because everyone knows that hip replacement work extremely well. That's been the subject of years of research, and everyone knows. You want to have a cataract done privately, that's fine. Everyone knows that that's been proven to be an effective therapy. If something is not known to be effective, then it should be the subject of a trial. If it's subject of a trial, you shouldn't be paying for it. That should be sponsored by either a company or a charity or some national funding agency. So I tell people, do not go and have a stem cell therapy, which you're paying for anywhere in the world.
Roger, thank you very much. I completely agree with that. I think Leah and I had a fascinating journey flying across America, and in the back of the In-Flight magazine, there were 2 ads for stem cell clinics, and I have them pinned up in the office just to remind me that actually it is important that stem cell tourism happens, but it shouldn't. It really
and if
shouldn't.
it goes wrong, it has an effect. So that, there was a child who had a stem cell transplant in Russia about 20 years ago, died of a tumor. There was someone in America who's had a spinal cord injury, had a stem cell transplant, now has a spinal tumor. So people do die of these therapies. I
think there's another question coming up from Suzanne online.
I think we can switch to in the room.
Oh, sorry, Claire.
Thank you, Andy, and thank you, Roger. Thank you, and thank you for the your tenacity. So my question pertains to 2 things. One of which is trying to choose the right age of your patients in the cohort, and secondly about the infusion-induced dyskinesias and your perception of how that might evolve with the next cohort of trials.
Yeah, so why did some patients develop these involuntary movements with the transplant? So the obvious answer would be that we just put in too many dopamine cells and they were overdosed. No evidence for that. What is thought to be the reason is that when you take the developing bit of brain that we use for those transplants, you have the dopamine cells, and next door to it you have another group of cells which are called serotonin or 5-HT nerve cells, and they get transplanted in, and it's those cells which then produce the dyskinesias. So it's a contaminant within what would be a natural contaminant within that midbrain preparation that we make from the fetal tissue, which is causing it. Now, obviously, with stem cell derived dopamine cells, we don't make serotonin. We know because we can look at it. So, we have absolutely none. We can't find any. So we think if that is the cause, then that would not be the reason. That would be our optimism as to why nobody would get a graft-induced dyskinesia. In terms of selecting age, it's always a very tricky one, really. And when we did a survey, when we did TransEuro about we're going to do fetal transplants, what are the worries that people have? We did it with Parkinson's UK, actually, and the biggest criticism we got was we had an upper age limit. This was regardless of fetal tissue, brain surgery, immunotherapy, no concerns about that. It was about why are you being so ageist? And it is a difficult question to answer. It's just that the clinical data so far suggests that people younger, so 50s and 60s, when they have the transplant, especially 50s, seem to do better than people who get it who are older. That's not to say it wouldn't work. It's just that's an observation that we've made.
I think we've got another question online, Suzanne.
Yes, I've had a number of people asking about DBS, deep brain stimulation, and stem cell therapy.
Yeah, so currently, if you've had deep brain stimulation, you wouldn't be suitable for one of the cell-based trials. And the reason for that is that you've already had neurosurgery. So that makes it more complicated to do a second round of neurosurgery with transplanting cells into the brain. There is also the unknown of what happens with a deep brain stimulator in the cells. I imagine absolutely nothing, but I don't know that for certainty. And the regulators would want to know that there was no interaction between those two. It doesn't mean you can't do it the other way around. So some of the people, for example, who had the fetal transplant trials who developed the dyskinesias were treated with deep brain stimulation, and it worked quite well. So you can have it, you just can't have it that way around. In the future, I think that this will become less of an issue. Obviously, if I'm right and they work very well, then you don't need deep brain stimulation.
Thank you, Roger. I think we've got another question in the room. Actually, I think David's got a microphone at the back.
Hello, can you hear me? Roger, two things. First of all, earlier in your talk you mentioned tortoises. You haven't seen me trying to get out of bed in the morning. I look like a tortoise on my back, quite frankly. Secondly, and more importantly, you gave an indication of some hideously expensive costs involved in treatment.
Yeah.
Can you indicate what the costs are, how they break down? What causes those big numbers?
Yeah. Well, there's the actual cost of making them. And then there's the cost of what they will be marketed at. And this is an issue, really. So, I mean, the cost for making them isn't that expensive. So it would be, I would think at most a few £1,000s. Obviously, there's the cost of the surgery and everything else, but it would be, in theory, it should be no more expensive deep brain stimulation. It actually should be cheaper because you won't need deep brain stimulation. You have to change the battery, and that's several £1,000s every few years. So it should be cheaper than that. It's very difficult to know because you have no control over the price. So, you know, a warning for us was that coming back to this monoclonal antibody for MS, which I told you took 22 years, when it was eventually approved, the company that then owned it at that point, it was already being used as antibody for treating rejecting organs, but because it was now indicated for MS, they took it off the market, repackaged it and brought it back on at 10 times the price. So the paradox was 10 years ago that despite Cambridge having done all the work to show that it worked in MS, one of the few places in the world you couldn't get it was Cambridge, because the NHS would not pay that price for that drug, which they knew cost much less than that. So it is a worry that the ultimate cost will determine whether you have this. And as you've been reading about recently, you know, these gene therapies for childhood motor neuron disease, you know, £2.5 to £3.5 million for that therapy. Now, it will never be that cost, but obviously the health economics of this will ultimately be a determinant. And a lot of cell therapies, not for Parkinson's, because I say they haven't been proven, have fallen down because the cost has put them outside of what people will pay for reimbursement for them. So it is a big issue, the cost. But there's no reason to think they should be expensive because they're not expensive to make, and you can make a lot very quickly.
Put it in context, they spend a fortune with failures and have to cover those costs of the failures.
They do, and what they'll say here is, of course, it costs a lot of money because we had to do all the development. Well, we did all the development. They've bought it at the end of the day. So
Fantastic. I think we've time for one more question online and one more.
There's a
from the room,
lady
if that's
here,
okay.
Helen.
Obviously really hopeful and share your enthusiasm for stem cells, but my husband was in the control group for Trans-Euro.
yeah.
He was obviously desperately disappointed not to have the treatment. And being from that situation, we're fascinated to know how many of those 11 people benefited. It's something I couldn't find online.
The Trans-Euro.
Yes.
Yeah, so I mean, it's not online, that's why. And the reason is that, I mean, it's taken a lot longer to analyse the data than I thought. The reason being that COVID, I mean, it was already a difficult trial to begin with, and COVID completely messed it up because lots of the patients came to the end of the study during COVID, and we need the imaging to correlate the imaging with their clinical response to know why people had improved or not improved. I think the sort of my take-home message from the patients I've seen with it is that there hasn't been a dramatic improvement, but they've actually stabilized quite significantly, which has been also what I've seen with the gene therapy trials as well. But I'm sorry about that, but I'm waiting for the imaging data in order to finish that off. And as soon as I've got it, then we'll be ready to publish that.
Thanks, Roger. Suzanne, is there one more online?
Yes. Excuse me, I'm sorry, I'm scrolling down, forgive me. Yes, a couple of people asked about GBA-corrected neuron cells.
Yeah.
And potentially this could address not just the dopamine deficit, but also the root cause of Parkinson's in patients with a GBA mutation. Could we have your thoughts, please?
Yeah, so I guess the idea here is rather than using a cell therapy, you just do a gene corrective therapy. Is that the idea? So people are trying this. So I think you can try and, so if you know a disease is due to a particular gene, or you want to actually edit a gene which is contributing to it, then obviously it's good to go after that gene. So the classic one, which we've been a bit involved with, is Huntington's disease, different disorder, known gene causes it, knock out the gene or knock out what the gene produces, and you've solved the problem. That is correct. But how do you do that? And how do you do that at every cell in your brain? And that is the big problem. So the science is there to do it, but to do it and deliver it is the big problem. And then the other problem with that is that if I edit out your gene have I edited out just that gene, or have I edited out something else? And even if that gene is a bit abnormal, is it still doing something that's good? So, for example, in Hunter's disease, you have one abnormal gene. It produces an abnormal protein. That's what causes the gene. I have a patient who has two genes, which have the abnormal gene. So both of his parents had Huntington's disease. He's in his 50s. He's fine. So his abnormal protein is keeping him alive. So it's not as easy as you think, but it is an area people are going after. And the capacity to edit genes and to knock out the intermediate, the RNA, with these things called antisense oligonucleotides or interfering RNA are in trials, but delivery is the big problem.
I think we've got time for one final question, which I think is John.
Hi, Roger. How do you reconcile the observation you draw our attention to, that many of these patients appeared neurochemically or behaviorally to be worse, and yet still felt fantastic? I'd be happy to take anything that would make me feel, would actually make me feel good.
Yeah, it's a good question, because there's one of these patients I used to have video of who was much, much better. And as a statistician, and I actually said, you can't say with a patient one why that's worked. And I say, well, no. He said it could be, it's a placebo effect. And I said, well, if that is a placebo effect, I don't really care. I mean, if I can make people back to normal 20 years after I've done something, just on the belief that they think they're better and I think they're better, that has got to be a therapy worth having.
You mean that's only time?
I think the problem is, so for example, it gets complicated, and I have to be very careful what I say here. But one of the cases I showed where the patient showed a dramatic improvement in their clinical response, they had actually contributed fairly significantly to the research program around that therapeutic. So obviously, if you make a major contribution to something, you're probably going to be seeing the outcome in a rather different way to how other people might do it. Although there was an interesting study, which was a little letter in a journal a number of years ago, where they looked at people's clinical response against the amount of money they'd spent on one of these stem cell clinics. And there was no correlation at all. So if you spent 15,000 or 5,000, none of the patients felt better. Obviously, it was a questionnaire sent out. So obviously the people who are likely to respond are going to be the people who are a bit cheesed off that they've wasted their money on one of those therapies. But it is a big problem, this mismatch. So deep brain stimulation, for example, often gives people fantastic clinical responses, but they don't necessarily feel fantastically better for it. And so trying to marry up what is a clinical response versus what is a meaningful patient perceived response is something that we need to sort of better understand.
Roger, thank you very, very much indeed. That really was a most fantastic talk. And it's given us such an overview of where the field is now and where the hope is for the future. So thank you very, very much indeed.